In this video, I'm going to talk a little bit more about crosses, but this time we're moving beyond the monohybrid cross. So monohybrid, remember, is a cross in which we're focused just on the passage of one gene from parental to offspring generations. And now we're going to move on to the dihybrid cross, which is now a focus on two genes from the parents down to the offspring. Put this into context, whenever there's a cross, you're actually passing on all of the genes from parent to offspring. Just for simplification, in a class like this, we only focus on the passage of one or two genes out of all of the genes that are possible. So let's move to this dihybrid cross. Again, this is looking at two different traits at the same time. And in the scenario I'll show you in a moment, trait 1 is going to be hair color, trait 2 is eye color. For these dihybrid crosses to work in this class, we're going to need to assume a couple of things. First, both of these traits can be passed on through genes. So hair color and eye color need to both be genetic. And the second big assumption is that each of these traits is expressed from a different chromosome. So we could say that maybe hair color is on chromosome 1 and eye color is on chromosome 5. The reason for this second is that it means that they can actually be passed on independent of one another. So you can pass chromosome, or basically an offspring can get chromosome 1 from mom and chromosome 5 from dad, that kind of idea. Okay, so the setup is hair color. Brown is going to be the dominant color over blonde. So brown is now going to be represented with the capital H, blonde is with the lowercase h. For eye color, green eyes are going to be dominant over blue eyes. And so green eye here is a big E, capital E, and blue eyes are a lowercase e. So to put that in a different way, here we have 4 individuals. Each one could be a parent from the P1 generation, and we know a few things about their genotypes. So if someone has brown hair, that person could either have a homozygous dominant genotype or a heterozygous genotype. If the person has green eyes, again, you could have homozygous dominant or heterozygous for eye color. For blonde hair, the only possibility is homozygous recessive for hair color. For blue eyes, the only possibility is homozygous recessive for eye color. Okay. So now let's think about all of these crosses. So here's several phenotypes. So here all we know is the phenotypes. Basically, we're going to do a cross and try and figure out what the offspring will look like. And if you were to guess that this is just a more complicated version of a monohybrid cross, you would be pretty much right. So dihybrid cross, we're going to start with just double heterozygotes because it makes the points the best. So again, here we have a person with brown hair, green eyes, crossed with another person with brown hair, green eyes. It turns out that there is a method that is used to try and make the creation of a Punnett square a little more straightforward, and it's called the FOIL method, and that stands for first, outer, inner, and last. And what do those basically mean? Our purpose here of the FOIL method, by the way, is to determine the sperm and the egg combinations. So here you can see at the bottom, we have our possible eggs and our possible sperm. What I really want you to note down here is that each egg contains just one copy of each gene. Each sperm contains just one copy of each gene. You don't have a big H and a little h in one sperm. It's not possible. So, or it shouldn't happen. Instead, you have one copy of H, one copy of E from or in each of the gametes. So FOIL. So first is first. That means that you're taking the first letter from each of the traits. And in this case, that's a capital H and a capital E. So that's first. Outer, you're now going to combine the outermost letters. So in this case, that's a capital H from the first trait and a lowercase e from the second trait. So there's our second combo. Inner, you're combining the inner two genes. So in this case, that is a lowercase h from the first trait and a capital E from the second. So there's your third possible egg. And the fourth is last, so it's the last gene from each. That's a lowercase h and a lowercase e. So there we've rounded out our four different possible eggs or sperm, our four different possible gametes. So now we're going to make a Punnett square from this. So what we want to do, remember, is cross these two individuals. We've got the mother and the father, so the eggs are these four possibilities, the sperms are these four possibilities based on the genotypes of the parents. And what we know is that we should end up with four different options for each in the dihybrid cross. And so basically what that means is we're going to need to create a Punnett square. Remember, we've got our two parents are both heterozygous for both traits. And so we're going to do that cross. It looks like the letter didn't get switched here. So F is eye color in this chart. Okay. With that being said, you can see here that the way I've done it, I've used the FOIL method. I've developed those four possibilities. So remember, there's just one copy of each trait in each of the gametes. So here up on top, let's say we've got the paternal, and on the left, you've got the maternal gametes. So sperm on top, eggs on the side. All right. So now that you've done that, you basically just do the crosses. So we have a capital H in this sperm and a capital F. So those are going to get added to the offspring. The other gamete here, the egg, is also a capital H and a capital F. So those get added. You just complete that through the entire table. This time it's going to be 16 squares in that table. And then you can answer all the questions you would need related to the phenotypes or genotypes of these offspring. So it turns out that this all represents the law of independent assortment. And I'll talk a little bit more about this with an example in the final video for this module. But basically, the law of independent assortment says that when there are two different traits on two different chromosomes, they're going to assort independently. I think you actually already saw this word when it came to meiosis. So just go back and look at the meiosis slides to think about the law of independent assortment. So what does independent assortment mean when it comes to chromosome splitting during meiosis? And then just think that there's going to be one trait on each of those chromosomes. It's a lot more complicated than that, but just go with that to help understand how it relates to inheritance. All right, so here's a much less exciting version of the same thing. This is basically stripping out all of the other stuff. And in this case, we don't know what these traits mean. We just know that both of the parents are heterozygous for both. And so we split them up again. We do the FOIL method. So basically, we do the first, so capital, capital. We do the outer, so capital, lowercase. We do inner, which is lowercase, uppercase, or lowercase capital. And then we do last, which is capital, lowercase. And so you can see that's happened over here. We formed our four gametes for the dad. So each of these represents one sperm cell. We've got four gametes for the mom. Again, each of those represents one egg. And we just do the cross. So basically copy down the letters from the parents, combine them, and you get the offspring. And you can use this table to answer any question that comes up related to dihybrid crosses. All right, so what I just showed you was a cross between two heterozygous individuals. However, you should be able to do this dihybrid cross with other types of crosses. So what I would recommend to you is just try to create a table like this and just type in the different possibilities. So we could have a parent, again, who is heterozygous. We could have a parent who's homozygous recessive for one and heterozygous for the other. We could have homozygous for both traits. Oops, come on now. There we go. We could have homozygous dominant for both traits. So all of these are possibilities. There are more possibilities as well. What I would recommend to you is just draw these 4x4 tables and try all of these different possibilities and see what the results look like. So here's an example where basically what we want to do is count a specific type of offspring, and that is the homozygous recessive offspring. This is going to be another cross of the heterozygous for both trait parents. So basically, I can do this FOIL method once again. So the idea here is let's start with the first, so AB, and it's the same for both of the parents. So I'll just put one of those in each spot. Outer is going to be A and lowercase, capital A, lowercase b. So again, there we go. Since both of the parents match, it'll be the same again. Now we do, I just did outer, now inner. So we've got lowercase a, capital B. All right. Again, it's copy, so the two parents are identical. We'll put it over there. And then finally, we have last and last, so lowercase for both of these. Okay, and again, the parents are identical in this case, so we can just copy it over. All right, so I have created the Punnett square, and now I just have to fill it in. So for this, let me actually try and make this a little prettier for us. There we go. So A, B, B, A, B, B, A, B, oops, this was a mistake. All right, A, B, B. Okay. And what I actually wanted was layout. Boom. Okay. So we're starting to fill it out. So A, B, B, A, B, B, okay. Looks a little different in that row. So now again, A, B, B. Finally, A, B, B, A, B, and A, B, B. Okay, so now we have our entire table filled out, but what is the question? The question is, what fraction of the offspring will be homozygous for both recessive traits? So it looks like there's only one. There's one possible offspring that is homozygous recessive for both traits, the one represented by A and the one represented by a B. So if there's just one individual, or one F1 individual, that means it is 1 out of 16 possibilities. And it does say what fraction of offspring. So there you go. 1 out of 16 are homozygous recessive for both traits. There you go. Let me get this back in there for you. So that would be the answer to a question like this. And hopefully that's helpful that I just walked through it for you. But again, I highly recommend that you do this for yourself. So basically, figure out what are the possibilities for the parents. So try one trait at a time. So again, for one trait, they can be heterozygous, they can be homozygous recessive, or homozygous dominant. And then you just make the combinations. So for each trait, that's the possibility. And now you can combine the two traits in all sorts of different ways for each parent. So again, just practice these over and over again with all sorts of different combinations for the parents. And then try and ask yourself different information about the offspring. So what portion of these are homozygous recessive for both traits? What portion are homozygous recessive for one of the traits? So in that case, it's a little more challenging to go through and count. I've seen mistakes basically where people skip over some. So here you've got one that's homozygous for one, but not the other. Here's homozygous for a second, so that's two. 3, 4, 5, 6, 7. So it looks like we've got 7 if I'm asking how many are homozygous for at least one trait. It's 7 out of 16. So that's just one other example. All right, so in the last lecture that's coming up next, I'm just going to go over the law of independent assortment again and give you yet another example.